This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs.
We have 5 experts from different fields of the primary research on dealing with cancer. We're going to talk about the similarities and the differences of the fields they all know. These are all 5 medical doctors. They're people whose work you probably have followed. Actually, I'm curious. We were discussing beforehand what kind of audience we'd have here. How many people just in this room have a medical degree of some kind? How many are involved in the bio business in some way? And how many have cancer as a focus of your work? So it's an obviously very well-informed audience. We're joined by many, many people who are going to be seeing us online. Over time, usually about 1 million people have downloaded these videos over the course of a year. So we'll try to give you our ambition over this next hour is to try to say from people who are leaders in differing fields of cancer research, what are the most promising developments they are seeing? What are some of the challenges they have, both in the science and the funding and business models and public policy? The areas in which their work overlaps, some ways in which it might differ, and finally, how we will think differently as informed citizens about this challenge that, as we've heard through the previous day's sessions, is so important. So I'll introduce the panel briefly. If I gave their full criteria and backgrounds, that would be the whole hour. Also, each one of them could fill an hour or two, but I'll just briefly say we have on my far left on the panel, Dr. Scott Lippman, who's the director of the Moores Cancer Center here at UCSD. He's a local San Diego guy from Grossmont High School. I was from Redlands High School, which was a tennis rival of yours, much earlier than yours, but you went on a tennis scholarship to UC Irvine, right, before you got into your pioneering work in personalized medicine. We next have Dr. David Sadava from the City of Hope Medical Center. Many of you may know him from his lectures on, I think it's called The Great Courses, where you're talking, I really recommend seeing them after you check the video of this session for fundamentals of cell biology, what we know about cancer, what we don't know. He's worked at Claremont Colleges and also a UCSD PhD. We have we have Christina Vori, who is the president and interim CEO at the Sanford Burnham Medical Research Institute, an active cancer researcher herself now, originally from Finland. We have Christopher Slapak, a Lilly Scholar and Senior VP of the InClone Systems of Eli Lilly from the Midwest, who's been doing a lot of his own research too. And finally, we have Gregory Sorensen, who is the CEO of Siemens Healthcare North America, former professor of radiology at Harvard Medical School, and a very active participant in the imaging business, which is so important in this field, and which Siemens plays such a role in. To mix things up, I'm going to ask my panelists each for a brief 2 or 3 minute overview of what in his or her particular field is most exciting and that they want to convey to you. I'm going to mix things up by not doing it in this kind of order. I'm actually going to start with Scott Lippman. So you can tell us first, although this is different from what we said 2 minutes ago. This is real-time adaptation.
I can see we went through this whole order, and
yes.
So, well, I think the most exciting thing for me is the issue of personalized medicine, precision medicine. The whole field of oncology is going through a transformation probably similar to what the internet went through in the early 90s. It's changing completely. The way we defined cancer before was based on the site of origin, breast cancer, lung cancer, whatever. Now we're moving away from that, or to include that, to defining cancers based on a molecular signature. Genomics is just the beginning. There's proteomics, metabolomics, and so on, because we know that may be more important information to direct therapy. So this is changing the way we do trials, changing the way we develop drugs to treat cancer. So it's a very exciting time, but it's going to be very challenging to identify these drivers. You've heard of some of them, for instance, the BRAF mutation, which was identified in melanoma and transformed that therapy. The therapy of that disease. You may not be as aware that the BRAF mutation occurs in other diseases. I've had patients with lung cancer that have that, and if you treat them with the BRAF inhibitor approved for melanoma, you can see dramatic responses. So we're looking now at redefining cancer based on pathways. Certainly the context and the site will be important. So it's a very exciting time, and we'll see a lot of changes. The whole system now is undergoing a revolution. The classic trial designs that we've all grown up with don't really apply here. We're going for high-risk, high-gain types of studies. The trials we've done in the past to approve drugs for cancer, you've heard, can be studies of thousands of people for 10, 20 years to targeting an improvement in survival of 6 weeks, for instance. I think those days are over. Now we can actually look into a tumor. We have the technology to identify what drives these tumors, what makes them tick, what their Achilles tendon is, and then develop drugs to inhibit that. So exciting time in personalized medicine.
And thank you for that admirable response under pressure lead into our discussion. Let me have one brief follow-up here, which is that for lay audiences, even informed lay audiences, it's often very hard to gauge the time horizon we're talking about. We're talking about things 2 years from now, 50 years from now, to the extent there are revolutionary changes as of roughly when?
Yeah, that's a great question, and I should have made that point. This is not a vision that we have that in 5, 10, 20 years we'll get there. This is happening right now in the clinic. We are seeing patients that are coming in that their disease is completely resistant. The disease has failed all treatments. We're doing genomic profiling, and if we identify a molecular genetic driver and use that drug, we're seeing dramatic responses. I mean, we've seen young people who have had their sort of farewell a party where there's no reception, where there's nothing else that can be done. They come in dramatic responses. So it's happening now in the clinic. This is not something, genomics is not something that we're hoping will develop in several years or 10 or 20 years, but we're just beginning. There's a long way to go.
Right, thank you. So, Christina Voy, for your own work, your own personal work in cancer research and the work at Sanford Burnham, what is the most exciting or significant distillation of what you're doing and seeing?
So I'd probably like to highlight maybe 3 things, and they get back to what Scott already introduced to us. It's a really exciting era for cancer research. On one hand, we are making really great scientific advances in the molecular classification of diseases. Just some 10 years ago, we thought that breast cancer is one disease. Probably know today that it's some 20 different types of diseases with each of its own causes and hence probably its own treatments. And thanks to the technological advances, we are getting to the point, as Scott outlined, we can take biopsies of tumors and do very detailed molecular level analysis thanks to technologies in genomics developed right here in town by Life Technologies, Illumina, other companies. And our hope is that when we do that molecular diagnosis, we can then match a patient to the right drug and right treatment for that molecular signature. So at Sanford Burnham, we work on 3 what I think are really exciting areas of basic research, how the laboratory research can enable doctors to take better care of patients today and in the future. On one hand, we try to find ways to match those molecular signatures to the right drugs. That's an active area of work for many, many research cancer centers. We also make advances in a couple of other interesting areas. One is a concept of tumor reoccurrence, or cancer coming back, sometimes years after treatment. And what we now know is that in every cancer, there is probably something what we call a cancer stem cell that originally causes that tumor to happen. And it turns out that it's probably that cancer stem cell that also may be the culprit why the cancer comes back after treatment. So you can think of this cancer stem cell or tumor-initiating cell sort of as a queen in a beehive. If you don't destroy that queen, the beehive keeps coming back. And it is that cell that we need to find in cancers and take it out. We are also making advances in understanding the process of metastasis, the spread of the cancer in the body. As you may know, in solid tumors, about 90% of cancer deaths are due to spread of the tumor, some other parts in the body outside of its primary location. What we now know is that cancer spread actually takes place very early in the process of tumor formation. Cancer cell spreads, remains dormant in other parts of the body, and sometimes a year later can get reactivated. So again, what we have to do is to find those cells, what activates them, and how we can destroy them later.
Great, thank you. And I actually have a master plan in the order I'm following here, which I'll try to reveal as it goes on. I'd like to ask Christopher Slapak now about the work you're doing with ImClone and what is most important to know about that.
Right, so just to pick up on the thread, the genomics revolution, as it's been applied to cancer, has really changed not only the understanding of the elucidation of the pathogenesis of the disease, the classification, but also as we begin to design new therapeutics for it. I have the privilege of leading the early development group for Lilly and ImClone. We have a portfolio of roughly 24 molecules in development for cancer. And these, our group partners with our discovery scientists, either out of the ImClone research laboratories or the Indianapolis-based, or here in San Diego, we have a group as well, to start the first human data set, the first in man studies, first in human studies, then to go on to develop data sets and clinical trials in the early phases of efficacy-directed trials. If you look at the past decade, the biggest advances have come from small molecule inhibitors, so-called targeted therapy, as well as protein-based biologics, monoclonal antibodies. Advances in both our ability to do chemistry and protein engineering and the understanding of the molecular pathogenesis. There are so many targets now to potentially pick from. I think that's one of the challenges we have in industry. What are the best targets? Because we can either likely design a protein-based therapeutic or a small molecule inhibitor to one of those targets. You can't do everything. There's not unlimited resources, so you must make choices. You must kind of prioritize what you think are the most likely targets to ultimately impact this very difficult disease. And then we must design a series of clinical trials to really answer that question in patients with cancer. So that's our challenge, but I think we have enormous tools and technology that have come to bear, including next-gen sequencing, including advanced diagnostics to really select patients who are most likely to benefit, to answer that question quickly, and really focus our resources on molecules we think that are ultimately going to make it and benefit patients. So it's a great time. We're trying to use all the technology and knowledge out there to get to decisions quickly so we can advance the most important molecules ultimately to get to the largest number.
Great, thank you. That's now Gregory Sorensen to talk about the imagery and sort of real-world machinery aspects of all this. What should we know about the way that imaging and other sort of physical devices are changing cancer work?
Well, I think what we're seeing, Jim, is an increased sense of the value of knowing about your cancer. And I would put that into a couple of different buckets. Certainly, imaging now and other tools are enabling us to know whether or not patients actually have cancer. So, for example, the National Lung Cancer Screening Trials showed that if you're a smoker or a former smoker and you have a CT scan of your lungs and you find cancer at an early enough stage, those patients who are screened have a 20% reduction in their cancer mortality, which in context is 4 times or more bigger than all of the advances in lung cancer treatment in the last 20 years. So it's huge, huge impact by knowing if you've got cancer or not. Then I know from my own experience with patients that there's tremendous tension about knowing, well, what's the status of my cancer? Where is it? Is it in my body? Is it everywhere? There's no more sort of sense of dis-ease, if you will, as when you've first got the diagnosis and then trying to understand, well, how bad is it? Is it spread everywhere? Is it local? Is it treatable? What's going to happen to me? I mean, and so knowing that by imaging tests or other tests can be very valuable. And then there's a sense of knowing, is my cancer responding? We know so many patients get treated with drugs that don't work for them. And if we can enable personalized medicine by actually knowing what's happening to the cancer, what its types are, what its features are, and then whether or not the drug is working. All of that, I think, put together says we as a system, I think, need to figure out ways to incentivize early and efficient diagnosis. Because if you go off on the wrong diagnosis, you do a lot of treatment that never really helps anyone. The most wasted healthcare dollar, and I know everybody talks a lot about cost these days, but the most wasted dollar is the one that you never needed to spend because you didn't have the right diagnosis.
Great, thank you. And now in the wrap-up overview mode, this is in punishment or in recognition for having done all these great courses videos over the years of putting things in perspective. We're going to have Dr. David Sadava say what else, how you would, the work you're most interested in that you do personally, and how you would draw together some of the comments from your fellow panelists.
Thank you, Jim.
Anytime.
I'm a laboratory scientist. I go in the lab and I do things. And the cancer I work on is called small cell lung cancer. That's different from large cell lung cancer. I think you can understand, well, some of them have small and some have large cells. But really, we can classify it as a special type of cell. It's called a neuroendocrine cell. This is a very difficult tumor. It's a tumor for which we have a molecular biography, but it's not clear as to what the targets for therapy are going to be. So the treatment for this tumor is the same as it's been for the last 30 years, and the prognosis is dismal. Here's what happens. Patient comes in and through various ways is diagnosed with this small cell lung cancer. Typically, the tumor is going to be treated with a cocktail of drugs that are the same drugs that it's been treated for the last 30 years at least. These are called toxic drugs. They're toxic both to the cells and, sadly, to the patient. The tumor exquisitely responds. It's a fantastic response, both in the laboratory and in the patient, to these drugs. The tumors shrink, and sometimes the tumor will go into remission. You can't see any tumor cells, but it's lurking there. There are some cells somewhere in the body. And months to, sadly, a year or two at the most after initial treatment and initial response, it roars back. And this time, if you try the same drugs on it, it doesn't work anymore. It's resistant. And many of you have heard of insects that are resistant to pesticides. These are tumors resistant to chemotherapy. Now, at that point, what we'd like to have is a molecular biography, as my colleagues have described, of the tumor that would say, let's try this next drug and see if we can target that drug that's unique to this, the target unique to this particular tumor. There isn't one for small cell lung cancer. So the prognosis is pretty dismal. People don't usually live beyond 2 years to 3 years after diagnosis. Some people live longer, but not very long at all. So what am I trying to do in the lab? I'll get cells or biopsy tissues from patients, and I'll try things to overcome resistance. And it's a kind of classical approach of pharmacology, drug research, to try to overcome resistance. And I'm trying to use things that I think are going to work. But I've done everything including collaborating with a Chinese medicine doctor. I studied a herb that is used widely in traditional medicine to treat lung cancer in traditional medicines. And you won't believe this, but at the molecular level, the cells respond the same way as they do to chemotherapy. So we're trying things that may work to overcome resistance. Is this an exciting new advance? I don't think so. I think the optimism is going to come when we know more about the molecular biography of this tumor. But right now, we're stuck with trying molecules and seeing whether they will work or not based on educated guesses.
Great. Thank you. So I deliberately asked all of you to a sort of positive toned question to begin with of what's most exciting in what you're seeing, and you informed us admirably about that. I'm going to ask now a question where I don't know whether there's a positive or negative answer. As a reporter, when I talk to people in other fields of basic research in the U.S., often they say the funding is stop and start. We could be doing so much more if we had, this kind of guarantee or underwriting. How should Americans in the world feel about the basic ecology of the cancer research system? Is cancer research sort of doing as much as it could given the fundamental limits of the disease and of science, or are there some frustrations that you all share as, if only this were different, then we could do so much more? Anybody who would like to answer that?
Well, I will defer to my academic colleagues. This is a very difficult time, certainly in academic medicine, to maintain funding for basic research. The pharmaceutical industry is absolutely dependent upon good relationships with major cancer centers, academic networks, and other clinical trial networks to really test compounds in a proper setting. So clearly, all the major academic centers are suffering right now from cuts in funding, and it affects our ability to do clinical trials and ultimately to compounds out to patients. So although the pharmaceutical industry is viewed as a deep pocket if you will, because they've been traditionally financially successful. I think our ability to partner with academic and medical centers, it's seriously been undermined, but I'll defer to my colleagues to comment further.
Yeah.
No, it's a great point. This is the worst time to put the brakes on this field because, as I mentioned, we're going through a very exciting, transformative period, but it's going to take a lot of work, a lot of resources. And so it is very difficult. The cuts at the NIH level are very, very difficult to deal with, particularly, I mean, at the Morris Cancer Center, we're very successful in getting NIH grants. So getting a lot of grants, you're hurt even more as these cuts go down. So it's a real problem. But I want to pick up on what you said about the academia-industry partnerships. I do think that is a major area we need to go. In fact, the NCI Advisory Committee has recommended that in a formal report about 2 years ago, to form academia-industry partnerships to develop drugs quicker. And this is a major initiative at the Morris Cancer Center. We're actually working with Sanford Burnham and Salk. We've formed an affiliation called it turns out that we're an NCI-designated comprehensive cancer center. We have clinical care. There are about 40 in the country, and there are actually 7 NCI-designated, the highest level of recognition, basic science cancer centers. 2 of them are right here in San Diego, Salk and Burnham, within 2 miles. So we've actually formed a formal affiliation. We're working together to try to take advantage and leverage resources in a difficult time to help patients. And we're actually working through that network to interact and partner with biotech. You know, this is the second largest life science cluster in the country. Yeah. So we have an incredible biomedical environment, and I think it's especially in this period of excitement and promise to leverage resources across these various centers and industry.
Yes.
I would really like to echo what Scott has said. I think the U.S. government, NIH, National Cancer Institute, have been major drivers for cancer research in the world, really. And I think, as we all know, we are facing really challenging times in that funding right now. The key is to continue to leverage resources, to collaborate within academia, with the industry. I think Sid in his Skype interview mentioned, I think, Stand Up to Cancer. That is one effort, how one puts really scientists from all over the nation together, really tackle unmet medical needs in the area of cancer, and I think that's the way to go.
And just to follow up on this, there are some fields of basic research where the scientists say they are being really seriously hampered by limits in federal spending. Are the NIH reductions, are they a serious limit on what you're doing, or are they a sort of annoyance in the annoyance field, annoyance category?
I would say that we ought to double the NIH budget again. You know, we did that more than a decade ago. It was a brilliant move. It drew people to America in a way that enabled science, and it gave hope to a lot of researchers that they had a future, and it attracted many of the brightest minds. It may not be possible in today's toxic political climate, but it's an idea that I think it's time to reconsider.
You know, I've been doing some work in Washington trying to help them to think clearly.
Good luck.
And it's my UCSD training.
Boy, talk about your long-term research project.
Yeah, right, exactly.
And there are 2 major conferences in cancer in this country. One is for the cancer researchers. It's called the American Association of Cancer Research. It's held every April. And there are about 15,000 people there, all cancer researchers, and 5,000 presentations. Imagine. It's a huge effort. And the other one is usually held in Chicago, almost always. It's the American Society of Clinical Oncology. It's held in June. And equivalent numbers. And there's not as much overlap as you would think. It's amazing. So we're dealing now with 25 or 30,000 individuals who are dedicating their lives to cancer research. And I thought it would be interesting if I snuck in 5 senior congressional aides to the American Association of Cancer Research, where I was presenting a paper last year, and I didn't want them to see my paper, but this was last April, and we snuck in early in the morning, and I walked them around, and I showed them the magnitude of cancer research, and I showed them the reality of cancer research. That is, behind all the numbers and the printouts that the people in Washington are getting, and the policymakers tossing around $1 million here and $1 million there, are real people dedicating their lives to this disease. And I thought for these people, it was very informative for them.
And did they act any differently after that, from what you could tell?
Several of them have told me since that they remember that it put a face it put a face. It put a face onto it.
Yeah.
I must say that our national organizations are very good at trying to explain to our legislators, who are the people really to make the decisions about funding, what the value of cancer research is. I think there is enormous public goodwill toward cancer research as well. So I'm optimistic. I would like to see the NIH budget doubled.
Yes, yeah.
So, and it's just to pick up on that, it's these exact large societies that are sending out messaging that's pretty alarming about trying to lobby and get to work through our local states to get NIH funding up. It's a serious problem. I don't know if you want to say crisis, but it's heading that way. And in fact, I was at a meeting, AACI, a meeting a few days ago in Washington. That's the Association for the NCI-funded Cancer Centers. And Kathleen Sebelius spoke at that. Actually, she was supposed to speak on Tuesday, but they moved it to Monday because they thought there might be a shutdown. And they were right. Anyway, so she came, and she actually talked about this. And the concern that she said is that we may lose the talent that we have in this country to go to other countries if we don't do something. So it's, I'd just like to pick up on Dr. Mukherjee's thing. This is our problem. Everyone in this room, everyone who lives in this country, we have to own this, and we have to make, we have to push the government to increase funding, particularly in cancer, which is really transforming.
And so I think I know what the answer to this will be, but I have to ask, would all of you be in favor of doubling the NIH budget? And would that be a widely held view within your field? On the factor of political storytelling, this actually meshes with what you're saying. The Atlantic held an event about 6 months ago, which I won't give any details about, but one of the prominent speakers was a very, very well-known conservative legislator. You see this person on the news all the time. And the interviewer asked this person, he was against spending in all fields except medical research for NIH, and why was that? And he said, with no self-consciousness, oh, we have a family history of this disease. So you can look it up. So I think that the fact that there is a sort of family history that we all are going to be involved in these diseases is very important. Let me now ask you something which goes a little bit against the grain of the thing you've been saying, but I think it would be on an audience's mind. You've been talking about all the fine distinctions we have to make about different kinds of cancer, moving beyond thinking, oh, just it's this organ, but it should be this causation. From the public's point of view, nonetheless, at what pace do you foresee different kinds of diseases seeming more curable or manageable? 5 years from now, is there a certain kind of widespread cancer that you think will be more manageable now, or it will all be on the individual sort of person-by-person level, do you think? Is there a category of cancer we'll soon start thinking as a chronic disease?
It's a little hard to predict. Look at where melanoma was just a few years ago. It was absolutely one of the most refractory diseases in the advanced state, and boom, molecular pathogenesis, we have new targeted agents, now we have immunotherapy that looks incredibly exciting. It's a little difficult to predict, and we have to understand cancer is hundreds of diseases as we're doing the molecular pathogenesis. It's not one disease. So across the spectrum, I think there's advances being made, and sometimes out of the blue, we have incredible advances in a disease that really looked very hopeless in the advanced setting. So,
yes.
So I think it's probably fair to say that there might be two types of cancers, sort of stupid cancers and smart cancers. And the stupid ones are those that have very few changes or mutations in them. And we can treat them successfully. I think chronic myelogenous leukemia is one of the success stories where we know the driver for that disease and get some remarkable results for treatment. That cancer tries to change in response to treatment, but we can still keep up the pace with it most of the time. And then we have another extreme of cancer that's, I think, very smart from the point of view that if it wants to survive, it finds ways to change and can have thousands of mutations. Unfortunately, lung cancers tend to be in that bucket. So we need to really find, I think, ways of treating the cancers, the stupid and the smart ones, and then really understand the smart ones. Why is it that they change so much? Is there something that we can address that prevents them from changing in response to treatment and becoming resistant, as was pointed out earlier?
Yeah. So are there any other answers on this question? I have another one. You are experts in your field, but you spend much of your life dealing with even the informed lay public. What do you find yourself having to explain most often to non-experts? What's the main difference in worldview between people whose research is in cancer and the rest of us, the thing that you wish that we understood?
I think that many people in the lay public believe cancer is a single disease. I think that's a very common misconception. I think people are also very hung up on the causes of cancer. And this is a field that I think really needs a lot more attention than it's getting now. Everyone who gets cancer wants to know why they got it. If you're a smoker, that's easy, probably. But for many cancers, it's quite difficult to tell why you got the cancer. Sometimes there's just spontaneous changes in the genetic material because we're chemistry. We're just made of chemicals that aren't perfect. But other times it's from something from the outside. But people really wonder about the causes of cancer, and it's hard to explain to them that there's a gap of our knowledge there. So those are the two things.
Yes.
I'd say that there's still a lot of mystery around cancer for a lot of people. One of the reasons that we're so excited about supporting Sid's book being made into a documentary to make it more accessible is there's still a lot of fear and questions in people's minds. And I think demystifying it, like his book does for those who have the time to read a 600-page book, or I think the high school students who will see Ken Burns' documentary, they'll start to understand this is, it is a complex disease, but it's one in which we have ways to attack. We're not powerless in the face of it. And there's hope. There's reason for hope for it.
Yeah.
So I would say it's both the expense and the time it takes to develop new drugs. I came from an academic background to join industry, and I was absolutely amazed at the resources it takes to move a molecule from the bench into the clinic to an approved agent. Independent organizations like the Tufts Center in Boston that study this, have estimated new drugs cost $1 billion, $1 to $2 billion. I mean, that's no joke. I mean, in some regards, that's not sustainable. Where do you go to the next level, 2 to 4 billion for a new drug? So I think the current expense and the time, 7 to 10 years, would be, not unheard of, not average. And it's just a very time-consuming, expensive process to really guard that you are delivering only efficacious and safe drugs to the general population. So I think it's a problem.
Yeah. So I'm just going to ask one or two more questions before I turn it over to the panel here, because I know you all have yours. An understanding by some members of the informed but non-expert lay public is that as other preventable causes of death are eliminated, infectious disease, accidents, or whatever, more and more you have a world in which everybody eventually gets cancer. Is if you live long enough, you get some kind of mutation that will cause a cancer? Is that a understanding shared in your world, and what are the implications if so? If we all sort of survive the saber-toothed tigers of yesteryear and influenza and all the rest, and people live into be their 70s and 80s and 90s, how do we think about a world where most people expect to get some kind of cancer?
Well, I think the key is to work towards a solution or a situation where we understand that cancer might be a very common disease in one way, but we would like to have the cancer to be a disease that you can live with, not to die from. We have made similar advances, I think, in the cardiovascular field, and can manage really the diseases. And that in some ways might be the more realistic goal, rather than finding this silver bullet, the magic bullet that is cure for all and really eliminates cancer altogether.
Yeah, and I agree completely with that. I mean, we will have some cures of some subtypes, but my belief, my hope is that we can convert most cancers into chronic diseases, like heart disease, high blood pressure, and so on. And I think that's a realistic goal going forward.
Yeah. The other big picture final question I'll ask you is, one of the major crises for American medicine, as you know, is simply its cost. And the innovations you're working on, do they continue the ramping up in the expense of medical care, or do any of them hold the prospect of reducing that?
So I brought a gizmo to show us, and after the earlier thing, one of the things we introduced this year was a cordless ultrasound, where this is the ultrasound and it shows up like on your screen. And while it's expensive, it reduces infection or complications from doing procedures without ultrasound guidance. And it's one of those things that now independent societies say, well, you shouldn't put a central line in without ultrasound guidance. So it's one of these short-term blips in expense that cuts the expense long-term. And I think one of the hopes of healthcare reform is that you'll reward systems for making those kinds of investments. And there is some hope that we're starting to shift to these long-term accounting horizons so that systems get paid for doing the right thing. In the past, they didn't want to spend the money because they didn't get the savings. And so the innovation in even the business model, I think, is starting to happen. And that's a really positive sign.
Yeah.
And I think that when you discuss personalized medicine and targeted drugs and the expense, there's this sense that we can't afford this field, even though it helps people. And you know, I disagree, and we're looking into this in a business model, because if you look at it, if you treat all of lung cancer with the same drugs, which we do, treating 90% of the people who get sick, the drugs may be less expensive, it doesn't help them, versus identifying the 4% with a certain alteration that and treating with a more expensive drug. I think we're looking at those kinds of models, but I think a lot of people, the public and doctors that aren't doing this, the knee-jerk reaction is it's an expensive drug, we can't afford it. But the whole idea is you're just treating individual people. Also, I think that we have to, and it made me think of this when you talked about the technology. I mean, the way that technology is advancing is if we can get this into the clinic quicker, it will save costs and so on. For instance, we didn't talk about this, but from my view, one of the most exciting transformative events in cancer is being able to do what's called liquid biopsies and sort of convert solid tumors into hematologic tumors where there have been major advances by monitoring free DNA in the blood, circulating tumor cells. The technology is getting rapid. Even here in San Diego, a lot of work. And so it's not out of the realm of thinking that in the not distant future, people will walk around and they'll have samples. And if a mutation appears, resistance mutation, they don't even have to come in. It'll pick it up. It'll get remotely translated. So I think the technology can really help us drive this down if we can get it into the clinic. And in this region, the technology with Qualcomm and so on is so strong that I think we can do it.
Great. Yes.
I think the key is really in the some pure cost perspective, as I think quality of life perspective. I think the key is really prevention, early detection. This is the absolute best way in the cancer field. But then wanting to comment a little bit what Scott said, a major cost is that we are treating patients with expensive drugs, and we know probably that these drugs will not work. We just don't have any better options available. So the cost of that treatment is tremendous. The cost of life, of working days, toxicity, side effects is again tremendous, and that is a very big burden cost-wise. And I think it's really up to us to find the ways to find the patients who will benefit from certain type of treatment, also find the patients that we can't and don't want to treat with certain type of medicine.
I think there's going to be a big issue going forward with translating a lot of what goes on in academic centers to community oncology. It was mentioned last night that 80% of cancer patients are treated not at an academic medical center, but the model in America is it's diffuse. That is, there are many community cancer centers. And the treatment is very different in those community-based centers. It's not as focused, it's much more traditional, and it's not as efficient in terms of use of resources. And hopefully, and this, as we move forward, the doctors out there in the community will become more informed about evidence-based medicine, which is really, it's not only a buzzword, but it's a reality.
Yes,
I was just to make a quick point on that, and picking up on Sid Mukherjee's comment about the percentage of people that go on trials. This is a huge problem. If you look at pediatrics, it's 80%, very high percentage of children with cancer go on trials, and there have been major advances there. This 10 to 15% of adults with cancer that go on is a real problem, particularly when it's known that people that go on trials actually do better. Yeah. So we need to somehow, as a society, get that message across and really speed things up.
So questions, if you raise your hand, a microphone will come to you. So yes, this lady back here, start off. And actually, why don't microphone bearers just find the next person, so I'll call wherever the microphone goes. Yes.
Hi, I'm Juan Dang. I had a question for Scott from one of your opening statements about clinical trials. How do you envision future clinical trials being changed from how clinical trials are run now, the traditional model? Because I think that's going to change. And how do you envision that?
Yeah, it's a very good question. And I don't know that anyone knows the answer. I don't know the answer, but to give you an idea, you may have heard this phrase, N of 1. There's actually a company, N of 1. There's the NCI is taking this on, the FDA. The concept there is that if you see a patient and the tumor doesn't respond to anything, you do genomics, you find a driver mutation that you know is driving that tumor, you show it in the lab, and you give a drug that inhibits that, and the tumor melts. And these responses can be dramatic. How many patients do you need to treat with that to know that you've got a good drug? to give them, and how do you do that? I mean, 1 patient, 2 patients, 3 patients. The standard way we develop drugs in the past has been to do phase 1 in broad, unselected patients, then phase 2 in 50 or more patients, and phase 3. I think you're going to need to do those studies for safety and others, but it shouldn't take that long and that many patients to know that you have a drug that can help patients now get that to them, and particularly if there's nothing else, and then go on to refine the development of the drug and dose. So that's what I mean by changing the trial designs. Doing selection in the earliest phase. We do it at Moores. We actually, in the earliest phase of drug development now, we're doing genomics and we're enriching certain drugs and certain pathways. So we're not doing it just randomly based on a refractory tumor. We're actually looking at genomics in the earliest phase because patients are benefiting there. Long way to go. So there's a lot of debate about this. We know it. I think the one thing everyone agrees with, I hope, is we have to change the trial design. The whole paradigm has to change. Yeah. That we agree on. Which way? There's a lot of debate.
So who has a microphone? So the microphone is back here? Yes. Yes. Good.
Good morning. I'm Catherine Phillips at the University of California, San Francisco. And we work on taking personalized medicine and figuring out how to improve patient outcomes. So I'm a health economist, and we're looking at sequencing technologies. Now, they're very exciting in terms of what they can do, but there's a lot of concern regarding how we're going to deal with the huge amount of information that we get from sequencing. Because when you sequence a tumor, you frequently need to sequence the individual as well in order to understand what the tumor sequencing means. And so it provides a huge amount of information. It's not $1,000, which people tend to believe. Payers are going, oh my God, we're going to have to pay for this. Clinicians are going, oh my God, how do I interpret all the information?
This to its question?
So to its question is, how do you see us dealing with the challenges of using sequencing going forward? And do you see that occurring soon, or is it going to take a while?
Yes. I can start it off. I'm sure that a lot of people have comments. So it's interesting, the cost. The cost, I think, won't be an issue. I just heard last night that the president of Illumina said that within 3 years or so, they'll be able to sequence the entire genome for $500. So it's coming down dramatically. So I think the cost is going to keep coming down, which will mean, to get at your real issue, from my point of view, is what are we going to do with those information? I mean, people are walking in, they can get direct-to-consumer genomics and walking in with huge data sheets. That's really the big issue. I mentioned BRAF mutations and some of the others, which are pretty well defined. You can look for it, you can find it's easy, it's a driver, and you can treat. But I think most of cancer, particularly solid cancer, is not going to be that situation. It's going to be very complicated genomic patterns that you can't just look at a mutation and say this is it. You need a whole field of bioinformatics, very high level. This is beyond statistics, specially trained people that can look at all the stars in the galaxy and come up with patterns. Very complicated. We have a very strong program actually in the Mesa, at the Venture, at our place at Moores, and with Cytoscape. We're actually taking this on. So I'll say where we want to go with this, and this is it's hopefully not too much of a dream, but to make it the most practical is if we could link and share data across all the genomics, really across the world, so that we could actually look at patterns and look at outcomes. So you look at these complicated genomic patterns, and you see how patients do with certain drugs. So a doctor in the clinic can search that database and say, boy, this is the pattern, and there were 4 patients in Australia, 2 in Brazil, and they responded, that kind of thing. That's going to take a little bit of time to get to, but we're thinking about doing that now because it is a huge challenge. The last thing I'll say is that as doctors, and I've been doing this for a long time, we're not trained in this kind of biology. You either go into the lab, or you become a clinical person, or there's some that do physician scientists. Now, I recommend all doctors spend time in the laboratory, whether or not they want to do that for a living, because you have to understand the biology enough yourself, and then to be able to tell patients about it. So there's a huge transition, even at the front lines of the clinics, with doctors understanding how to do this and then reflecting to patients. So a lot of challenges.
Some brief follow-ups here? Yes.
Sure. So I think your question was really about 2 things. One is the cost, and that was already addressed. We hope certainly the cost of genome sequencing to go down. The other is the complexity of the data. How does a treating doctor practically use this data? And I think where we are really heading is that it's not an individual doctor anymore taking care of a patient, but we need to have teams of individuals, a tumor board, that brings expertise from the clinical medicine, the geneticists, bioinformatics, informatics people who interpret data. And it will be the consensus recommendation and decision by this team of experts really that end up making recommendations how to make the genomic data actionable and what is the drug of choice for this very specific patient.
So microphone on this side. Yes.
Hi, Rob McRae. And so I'm going to ask you a policy question. I've been struggling, all of you agree that we should double cancer research. And I'm thinking, where would NIH move the dollars from to double cancer research? And maybe is the solution to move it from health care with ineffective cancer therapies, something we all agree on, and look for the additional funding there?
Who has a view on that?
You know, I'll speak up. I think it's very true that we spend a lot of money on ineffective cancer therapies. And there's been tremendous work trying to identify who would benefit and who would not benefit from a given therapy. I went on an NCI panel a couple of years ago looking at that. And at the end of the 2-day session, trying to identify biomarkers that would predict who would respond to, at the time, bevacizumab, which was then the most expensive drug. The physicians basically said, look, unless you can tell me with more than 99% certainty that this drug won't work, I feel ethically obligated to give it to my patient. They've got no other options. And even though it's $10,000 a month, I've got to do it. And I think we're in a world where we don't really ethically feel like we can say no to certain things. And of course, that's a mythical world. Because we don't have the resources to keep doing that. But it's less about how do we make policy decisions about where do we do things and more about how do we start having hard conversations with ourselves about the limits. And as one of the earlier speakers said, we don't seem to pay for our own health care. And so that puts all kinds of weird misincentives into the marketplace.
Yeah, exactly. I think at a societal level, it's easy to say that. At an individual patient level, that's almost impossible to do. And you look at where the majority of our healthcare costs, they go to end-of-life care across all diseases, not just cancer. So I don't think we should single out cancer. But yes.
Before I go to the upper tier, there's a lowbrow parallelist in the publishing world. Everybody knows that half of all advertising is wasted. Nobody knows which half. That's how we say in business. Yes, up there, upstairs.
My name is Mary Ann Nice, and I'm a nursing consultant. And I have a question. I'm very connected with the lay population through friends and family, actually, with various cancer incidents. And although the discussion always goes to what can I do pharmacologically for my cancer in looking at molecular structures, et cetera, the conversation always goes to alternative integrative complementary measures as well. What can I do? Do I need to be looking at my diet in terms of alkalinity versus acidity, looking at animal-based diets versus plant-based diets? What can be done? Is dehydration a part of what's going on with my problem? And so I'm just wondering in terms of kind of where alternative complementary and integrative medicine plays in terms of the treatment, not as an either-or kind of thing, but together joining that.
Yes.
Almost all cancer patients do some sort of alternative medicine. Now, you have to define that very broadly, and the way they define it in government circles is that includes prayer. It includes a lot of things, but it also includes herbs and other things. Got to be careful about what's done. And one of the good things at NIH was they set up, National Institutes of Health set up a institute for complementary and alternative medicine. It gets over $100 million a year in funding. And they do actually research on a number of the things that you have actually mentioned. And the research gets published, and they try to evaluate whether these things will work or not. It's very difficult when you have cancer because you're faced with a very dramatic, life-threatening disease. And of course, it's natural for people to look up the internet and say, oh, there's this herb, or there's a guy in Texas, or whatever. I think that it's up to oncologists and physicians, the medical community, to use evidence-based medicine again and say there is evidence for this, there is evidence against this, or there is no evidence at all.
Yeah, I'm a believer in different types of alternative medicine. I didn't used to be, but now that we're taking this, I think the key thing is we want to do the same evidence-based work to prove it for people as we do with other kinds of drugs. And to give you an example, there's some recent work from Liz Blackburn at UC San Francisco, won the Nobel Prize for her work in telomeres, that actually shows in the setting of stress, chronic stress, and psychosocial stress, that if you can reduce that in a very rigorous way, your telomeres become longer, and short telomeres are associated with cancer, heart disease, and aging. So I think that I think there's a lot there. We just need to study these kinds of things in the same way.
There are a lot more questions. We have time just for one, which will be over here.
Yes, my question was, prior to discovery of some malignancy, is there a preventive procedure that can be undertaken to predict?
Oh,
Yeah.
BRCA1.
Yeah, there are, and there are many examples. One of the most dramatic effects of advances in cancer treatment, cancer control, has been the Pap smear and the cervical dysplasia, treating patients that way. So identifying premalignant lesions and treating them. Same things with colorectal adenomas, which aren't cancer. Colonoscopy reduces mortality. I mean, there are a lot of examples, and I think what you were getting at in addition is that we can take these genomic molecular tests to determine which of these premalignant lesions really are at risk to go. Many of them will just go away or never progress to cancer. So moving the genomics earlier in early cancer and premalignancy to help define which you need to treat in which ways is a very active area. Good question.
Yeah, the BRCA genes, for example, genomic markers. So we're beginning to elucidate them. There are not a lot of them, but.
So the test of a panel like this is that when it's over, you know some things you didn't know an hour earlier, and there's a lot more that you want to know. I think by that standard, this has been a tremendous success. Please join me in thanking our panelists.